When a bone drill touches the femur of a 3,300-year-old mummy, it sparks a debate that reaches far beyond the laboratory. Extracting DNA from the ancient dead is one of the most scrutinized processes in modern science. Intact DNA degrades over time. Inside an ancient mummy, the probability of finding pristine, readable genetic code is statistically close to zero. Even when material survives, it is often compromised. Modern human handling has contaminated the majority of historical samples available to us today. Researchers have found that authentic ancient Egyptian DNA aligns closely with historical populations of the Mediterranean and the Near East, a profile that differs from the modern people living along the Nile today. For centuries, we could only study their physical remains. We could scan their skeletons and map their artifacts, but their core biology remained inaccessible to the eyes of science. In Egypt, the environment itself works against preservation. Searing desert heat bakes the DNA. The high humidity inside sealed tombs rots it, and the sodium-based natron salts used in mummification shred whatever genetic material manages to survive. The breakthrough came with next-generation sequencing and polymerase chain reaction, or PCR. This molecular technique allows scientists to take heavily degraded, microscopic fragments of DNA and multiply them into a massive, readable sequence. PCR provided a mechanism to verify the biological record directly, moving past the constraints of interpreting physical artifacts alone. In 2010, the Journal of the American Medical Association published a genetic study focusing on the lineage of King Tutankhamun. The findings detailed a family tree shaped by inbreeding. The data showed that Tutankhamun suffered from Kohler's disease too, a painful bone disorder, and carried the genetic markers for multiple strains of severe malaria. But those clinical findings were quickly overshadowed by public controversy regarding the R1b haplogroup. During a television documentary covering the project, a split-second shot of a computer screen was broadcast. Viewers reverse-engineered that unverified frame, pushing unscientific claims that the pharaoh possessed Western European ancestry. Y chromosome DNA is incredibly fragile. When Howard Carter opened the sarcophagus in the 1920s, he and his team worked without gloves or masks, leaning directly over the royal remains and unknowingly introducing their own DNA to the sample. Academic certainty requires published, peer-reviewed data, not artifacts caught on television. A single mummy, handled by thousands of people over nearly a century, is too compromised to define the genetics of an entire civilization. To get a true picture, researchers needed a broad, tightly controlled sample. They found it at Abusir el-Meleq, isolating the remains of 151 different mummified individuals. Published in 2017, the Abusir el-Meleq project achieved the first highly reliable, full-sequence mapping of ancient Egyptian genomes. Over 1,300 years, despite conquests by Greeks, Romans, and Arabs, their genetic structure remained remarkably stable. Data shows their closest relatives lived in the Levant and Near East. However, modern Egyptians diverge, sharing 8% more sub-Saharan African ancestry than their ancient counterparts. This shift occurred within the last 1,500 years. It was driven by improved mobility along the Nile River and the expansion of the trans-Saharan slave trade. Population-level DNA proves the Egyptian civilization could absorb foreign political rulers at the top without altering the genetic makeup of its working population. At the macro level, this genetic reality intersects with a highly charged historical debate, the ancestry of Cleopatra. The historical consensus is consistent. As a member of the Ptolemaic dynasty, she was of Macedonian Greek descent, with minor Persian ancestry recorded in her lineage. Applying modern concepts of race to antiquity is a scientific and historical anachronism. The ancient world simply did not categorize people using those metrics. To see what late ancient Egypt actually looked like, we look to the Fayum mummy portraits. These paintings offer direct visual evidence of a diverse, cosmopolitan society. By the Roman era, Egypt was a mix of Greek, Italian, and native Egyptian peoples. They were united by a shared climate and culture, not by pure bloodlines. DNA reveals a society that remained genetically stable for millennia, while evolving into a culturally diverse hub of the ancient world. This leaves researchers with massive open questions. Will archaeologists ever find an unlooted Ptolemaic tomb to definitively sequence? Can we reach back far enough to sequence the pyramid builders of the Old Kingdom? And how will new isotopic analyses of their remains further rewrite the timeline of human migration? We are finally reading the biological code of the people who built this empire. Does knowing their exact DNA alter how you view the legacy they left behind?